diving into biochemistry a little more deeply now. We're going to take a look at kind of the beginning part of this material. As I've said, this is a unit that has a lot of information to it. It has a lot of concepts and things, so we're going to take our time and go through this together. You're going to want to pause, rewind, rewatch this video as we go forward. Just a reminder, obviously, with our e-learning virtual lectures, make sure you guys are taking notes in your written notebooks just like you would in class. Put your cell phones and other devices away just like you would in class. You can pause and rewind when necessary. There's going to be some embedded questions on Edpuzzle. Remember, those are always just a completion grade. And then also, any other questions that you have that you want to ask me, you can send via Canvas or ask during class time, which would be, of course, on Zoom or in our face-to-face classes. So, as again, you can see our main ideas here. We're going to talk about the importance of carbon to start with. So that's the first thing that we're going to look at here. Carbon is something that has set up an entire branch of chemistry called organic chemistry. But one of the first important topics that we need to look at, and you're going to see this down the road on quizzes and tests, not just this year, but in future science classes in IB. So an organic compound, first of all, is going to be a compound that contains carbon. So make sure you have this down in your written notebooks. It has to have carbon, all right? But the important part, the thing that is usually somewhat confusing for students, is just because something has carbon in it, just because its chemical formula includes carbon, does not mean that it's necessarily organic. So the first requirement for something to be considered an organic compound is that it contains carbon. So you're going to see that carbon atom, all right? But also it has to be found in living things as well, all right? That's extremely important, all right? When we say found in living things, it has to actually be a part of, or has to make up the composition of a living thing. All right, which means that it's not something that is just passed through a living thing or that a living thing uses, but it has to actually make up the composition of a living thing. All right, and this is where you can see the exceptions. Okay, for example, things like carbon dioxide. All right, if I ask you, hey, is carbon dioxide organic? It's just a natural trick question, right? So you say, well, it has carbon, so okay, yeah, it's organic, but it's actually not, because it does not make up the composition of a living thing. Think about that for a moment, right? The process of photosynthesis and cell respiration, right? Photosynthesis takes in carbon dioxide from the atmosphere, all right? And then it's going to help to convert that into oxygen, which is going to later be released by that plant, right? But that carbon dioxide does not make up the specific part of a living thing. It just passes through it. So the first big idea from this lesson is to make sure that you understand that an organic compound is a compound that contains carbon, but it's found in living things, and more importantly, it makes up the composition of living things. Moving on the lecture here, I'm going to have you go ahead and give 2 examples of an organic substance and then 2 examples of an inorganic substance. All right, and I'm going to have some multiple choice questions here for you to answer, just to make sure that you understand the difference between an organic substance and an inorganic substance. So take a moment and let's answer those questions. All right, so lots of examples of organic substances. Again, remember, it's got to be something that makes up a living thing. So, for example, DNA would be a good example of an organic substance, or simple table sugar that you would find, just regular sugar. And interestingly enough, the reason that table sugar is organic is because that would be glucose, right, which is one of our macromolecules, which helps to make up the composition of a living thing. And then actually sodium chloride, table salt, is an example, a good example for you of an inorganic substance. Simply, it doesn't have the first qualification, which is to be composed of carbon, but it's a very similar substance to sugar. So just keep that in mind, guys, as you're going through this, as I'm quizzing you on differentiation between an organic substance and an inorganic substance. It's very important to understand that carbon is super important. We're going to dive into that in a moment, but understand that for something to be organic, again, number one, it has to have carbon, right? And then number two, it has to actually make up the composition of a living thing, right? And of course, DNA fits that category. All right, moving on. So why is carbon so important to life, right? Why is it so important? Well, there's going to be a lot here, okay, that we're going to look at. All right, so the importance of carbon. So there's an entire branch of chemistry called organic chemistry, and this is something that if you go on and get a biology or a pre-med degree or a chemistry degree, you'll take an entire year of organic chemistry. All right, so that's what we are going to be looking at in this unit, okay? Organic chemistry is basically the study of any compound that contains carbon, all right? So why is carbon so important? So it's so important that we have an entire branch of chemistry that is devoted to it. It's so important that if you're going to get a biology degree, you're going to take an entire class on carbon, an entire class on organic chemistry. We're doing an entire unit on organic chemistry right now, okay? Well, to start with, from a chemical standpoint, the chemical makeup of carbon is very unique, all right? Meaning that carbon is able to bond with up to 4 other atoms, all right? So it has, basically carbon has 4 electrons that can form 4 covalent bonds. So if I look at carbon on the periodic table, I would see that it is carbon 6, okay, meaning that there's 6 protons and 6 neutrons in the nucleus of carbon. And then the way this works is in the first shell of electrons, there would be 2 electrons there. Let me change my colors here for you guys. All right, so in my first electron shell, it can hold up to 2. So there's my 2 electrons in the first electron shell. Remember, the atomic number here is going to be 6. Okay, so that means there's going to be 6 valence or 6 electrons in the outer orbitals of our carbon atom. My next electron shell is going to house the rest of these electrons, right? So we've got 1, 2, 3, 4, 5, and 6, getting us to that number of carbon 6, okay? And then at these locations now, right here, right here, right here, and right here, as we learned when we learned about the properties of water, we learned that covalent bonds are a sharing of electrons. So another atom can come here and bond, can come here and bond, can come here and bond, and can come right here and bond. So that chemical makeup is extremely important. So it has the ability to form up to 4 strong bonds. All right, so the bonding that is important to understand. So first of all, we have those 4 available bonds, which makes carbon extremely important. All right, so it can bond, carbon can bond with other atoms, such as nitrogen's an important one, oxygen, hydrogen, phosphorus, and sulfur. These are going to be the main bonds that carbon is going to form, especially in organic chemistry, or in what we're studying right now, biochemistry. Those are the atoms that we're going to see carbon bond with, okay? But along with this, another really important idea is that carbon has the ability, so this is important, carbon can bond with other carbon atoms, all right, and it has the ability, these carbon-carbon bonds can be single bonds, they can be double bonds, all right, so a single bond would look like this, a double bond would look like this, or a triple bond, which could look like this, all right. So these are the types of bonds that we can form, and because of all of this, okay, you guys know I'm all about big ideas, so because of all of this, carbon can form long chains and ring structures. All right, and this is where we get things like long carbohydrate chains or long fatty acid chains. All right, so that's kind of what we're looking at. So for carbon bonding, we've got it's a bond with atoms such as nitrogen, oxygen, hydrogen, phosphorus, sulfur. Carbon can bond with other carbon atoms, form single bonds, double bonds, and triple bonds. Carbon can also form long chains and ring structures. All right, the first ring structure that we're going to be looking at is going to be our glucose molecule. Okay, glucose is going to be one of the primary monomers that we're going to be talking about as we move our way into the macromolecules next. But to start out with, I wanted to make sure that we had a clear understanding of the importance of carbon, why carbon is such a big deal. All right, and now we're going to move into the biological macromolecules. The first concept that we have to understand, guys, is the difference between a monomer and a polymer, and how monomers can combine to form polymers, and how polymers can be broken down to form individual monomers. Here on the Ed Puzzle lesson, I want you to take a moment and answer the next couple questions. I'm going to give you some choices. I want you to decide which would be a real-life example of a monomer, and from the choices, which would be a real-life example of a polymer. Hopefully this will help you to start to wrap your mind around what a monomer is and what a polymer is. All right, now that we've taken a look at the basics of what a monomer is and what a polymer is in real life, we're going to continue on and look at these examples in biochemistry. So in your notebooks next, you're going to want to create a full-page table, kind of like we did before with a couple other concepts this year. So create a full-page table in your written notebook. Go ahead and make it landscape style. And we're going to follow this format. All right. So go ahead and take a moment and just transpose this table into your notebooks. Again, go ahead and make it landscape style so you have plenty of room. You can see that the first column are 4 macromolecules. These are the macromolecules that make up the composition of a living thing. The next column over would be, we're going to be identifying what the monomer is. So for carbohydrates, carbohydrate would be the polymer. Lipids would be the polymer. Proteins would be the polymer. Nucleic acids would be the polymer. All right. The monomer then is going to be considered the building block, as we've looked at. So it's the building block of that specific polymer. Okay, so the monomer for a carbohydrate is simply going to be a simple sugar. And we will get into examples that would fit into that. Okay. In fact, let's just jump over to the examples first. So my first example is going to be glucose, which we've already talked about. The chemical formula for glucose will be C6H12O6, meaning there are 6 carbon atoms, and we saw that in the ring structure. So glucose has this ringed structure to it. And remember, because of carbon's unique ability to form bonds and form structures, we're able to have this ring structured formation here. So our monomer is going to be, make a little more room here, our monomer is going to be a simple sugar, otherwise known as a monosaccharide, which means one sugar. One means mono, right? We all know that. And then saccharide means sugar, okay? And then all of these carbohydrates are going to have a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. And as you can see here with my glucose example, you see just that, right? We've got C6H12O6. That's a 1 to 2 to 1 ratio. For each one of our macromolecules, there's going to be some type of a chemical test that we're going to do to figure out if it is a sugar or not. So we're going to take a substance. You would have seen this in the identifying nutrients gizmo. You take a substance, you put it into a solution, you do a chemical test on it to see if there is sugar available, okay? So for carbohydrates, there are going to be 2 chemical tests that we need to know, all right? The first one is going to be what we call the Benedict's test. All right, and this is going to be to test for simple sugars. And when I say simple sugars, I mean just something like glucose over here. This is a simple sugar, all right? The second chemical test, and there are 2 that we need to be familiar with for carbohydrates, would be the Lugol's solution test. So Lugol's is just iodine, all right? So we can mix whatever substance it is with iodine, and then we can identify if it is a complex sugar. So when you would do this test, you would first do the Benedict's test, see if it's a simple sugar. If it's a simple sugar, then you could move on to the iodine test, and then that would confirm if it's a complex sugar or not. But obviously, if it's not one, it's going to be the other if it's a carbohydrate at all. We should all have a pretty good understanding of the function for carbohydrates. The big thing here is that this is going to be short-term energy, all right, in animals. So that's going to be in animals. And then in plants, it's going to help the structure in plants. And that's basically what helps to make up the cell wall and helps to form the structure of a plant would be a carbohydrate. All right. For my examples over here, I've got my simple sugar or my glucose. I'm also going to have polysaccharides, and there's going to be a handful of polysaccharides that we're going to have to be familiar with. All right, and we will be getting into those a little bit later. All right, that's our first group. That's our carbohydrate group. My second group is going to be a group of lipids. There's going to be a basic chemical test for the lipids, and that's going to be the brown paper stain, basically, because that's what we will call this, the brown paper stain test, okay, because lipids do not mix with water, so you can use a brown paper stain test, and that's going to identify it as a lipid. Our function for lipids, this is going to be more of our long-term energy, all right. It's also going to provide insulation as well. It's going to provide protection for the soft organs. The monomer for, or the building block for lipids, is going to be fatty acids, a fatty acid chain, and a glycerol. So the building block for a lipid is actually twofold. It would be a fatty acid chain and a glycerol as the building blocks or the monomers. All right. Examples for lipids are going to be fats, oils, waxes, or hormones. Those are all fats, oils, waxes, hormones are all examples of lipids. Now, we started by talking about the importance of carbon, okay? I'm going to put in red here the atoms that make up each one of these groups. So a carbohydrate atom is going to be made up of carbon, hydrogen, and oxygen. And lipids are also going to be made up of carbon, hydrogen, and oxygen. So those are two important things to recognize there. Moving on to our third group, we're going to be looking at the proteins, all right? And the proteins are going to be made in terms of the atoms that make up proteins. We're going to have carbon, hydrogen, oxygen, and nitrogen as well. All right, so nitrogen is something, if you see a molecule that has nitrogen, carbon, hydrogen, and oxygen, then you know that it would be a protein. The test for this is going to be the Buret's test, and we really, guys, just need to know what is the test. You don't need to know yet how to apply that test. The function of proteins, big structural function, okay, the structure, and then also it's going to have a function with enzymes, and we'll be learning about proteins and enzymes and how they work together as we go forward. But for structure, we're talking about muscles. Proteins build muscle. We're talking about muscle contraction, okay? And then for enzymes, most all enzymes are made of proteins. So proteins and enzymes work hand in hand together. Let's write that down, that all enzymes are made of proteins. Okay, just say that they all are to keep it simplistic. The monomer of a protein is going to be an amino acid. So we can take an amino acid chain and we're going to be able to form that into a protein chain eventually. Okay, examples, well, enzymes are the biggest example. Hemoglobin, which is in the blood. Hemoglobin is what transports oxygen in the blood. Insulin is a protein, believe it or not. Insulin is what regulates blood sugar in the body. Okay, moving on to nucleic acids, our last group. Well, first of all, for nucleic acids, the building block or the monomer for nucleic acid is going to be what we call a nucleotide. And let's focus again on the atoms that are going to make up a nucleic acid, right? So far we've got CHO, or carbon, hydrogen, and oxygen for the carbohydrates. For lipids, we have CHO, carbon, hydrogen, and oxygen. For proteins, we have CHON, carbon, hydrogen, oxygen, and nitrogen. And for nucleic acids, we have CHONP, like chomp, but CHON with an N, carbon, hydrogen, oxygen, nitrogen, and phosphorus. The big examples here are going to be DNA and RNA, so all of the genetic material. So DNA and RNA, and for our function, we're just going to write that this is the genetic material. Again, as we've talked, this is organic, simply because that's what an organism is made of. Okay, so all of our genetic material there, and then we're going to take a look at the test, and there really is no test because all living things are going to have some form of DNA, so there is not actually a test to test and see if a substance has nucleic acids. So as we wrap up this lesson on our kind of an introduction to biochemistry, here is what you should know as of now. We should understand, first of all, the chemical importance of carbon. We should know what organic chemistry is. We should know the difference between an organic substance and an inorganic substance with examples of each. Okay. We should also know the difference between a monomer and a polymer. We should know our 4 macromolecules, which are carbohydrates. If you hear the noise in the background, that would be the dog sleeping. He's not too excited about macromolecules, apparently. We've got carbohydrates, lipids, proteins, and nucleic acids. All right. We should know that for carbohydrates, the monomer is a simple sugar. For lipids, the monomer is a fatty acid and a glycerol. For proteins, the monomer is an amino acid. And for nucleic acids, the monomer is a nucleotide. So make sure you're taking time to review those. Okay, the last thing then is going to be our macromolecule chart. You should know everything from the macromolecule chart. We will be having a quiz on this material. As you might see, I did not get to the macromolecule reactions yet because I don't want to rush too far ahead. But the next lesson that I'll be posting will be on the macromolecule reactions. And if this is our 2020 class, that will be one of the first lessons right after your Thanksgiving break. All right, so there's your summary. Keep an eye out for the quiz popping up on Canvas on this material. Again, if this is my 2020 class watching this, you'll need to have that quiz completed before the end of the week. Thank you.